Meiosis has two major divisions, which lead to 4 cells that have a haploid number of chromosomes. It starts with the diploid cell, an interphase, where DNA replication will occur. Then meiosis 1 can begin, splitting the cell into 2. The cells don't replicate their DNA before the next division. In meiosis 2, the chromatids separate into chromosomes, leaving 4 haploid cells. These haploid cells will mature into gametes, sperm or egg. Let's look at this process step by step in more detail. During interphase, the DNA is replicated in preparation for meiosis 1. The chromosomes aren't actually visible yet, but I'm showing them as chromosomes here so that it's easier to see that we'll follow a pair of homologous chromosomes through the process. The first step of meiosis 1 is prophase 1. During prophase 1, the nuclear membrane dissolves and the centrioles move to the poles. But something very strange happens with the homologous pairs of chromosomes. The duplicated homologous chromosomes are called a tetrad. And when the tetrad pairs up, it's called synapsis. Once they're paired up, they will recombine their genes by crossing over. The duplicated chromosomes are no longer identical, which creates variation and diversity in the final gametes. Spindle fibers will attach to the chromosomes, and they'll move and line up in the center during metaphase. Now, which side they face is random and is also known as independent assortment. They will not be pulled apart at the centromere like in mitosis, but instead, homologous chromosomes will separate. At telophase 1, the nuclei reform, and the chromosomes wind into chromatin. And during cytokinesis, the rest of the cytoplasm divides. In humans, there's no second interphase. However, some plant species experience a second interphase, or interkinesis, but the DNA is not duplicated in this phase. During prophase 2, the chromosomes reform and the 2 nuclei break apart. Then the centrioles send out spindle fibers and connect to the centromeres of the chromosomes. During metaphase, the chromosomes line up at the center. This time, it's in the same direction as it would be in mitosis. Then in anaphase 2, the chromatids separate into chromosomes as spindle fibers pull them to each side. In telophase, the chromosomes unwind into chromatin, and the nuclei reform. Then the cytoplasm divides in cytokinesis, and we're left with 4 haploid cells that are all different. Again, those differences are because of crossing over and recombination. Let's look at the process of crossing over one more time. These duplicated chromosomes are homologous. One came from the mother, and the other came from the father, but they both carry the same type of information. During synapsis, sections of the chromosomes will cross and switch, creating recombinant chromosomes. This provides a great source of variation.